Hello, everybody. My name is Rifaa Qadri. I'm a first-year master's student here in the Computer Science Department at the University of Maryland in College Park.
Hello, everyone. My name is Faizan Wajid. I am a fifth-year PhD student here at the University of Maryland Computer Science Department. Also here to represent is Marakai and Professor Ashok Agarwala, who is a supervisor of the Mind Lab. To describe our work here, it's titled Breath Analytics. The unique breathing pattern of an individual can yield a wealth of information. In fact, the 24-hour activity supports life by continuously driving gas exchange with incredible efficiency for up to 1 billion breaths in a human lifespan. With the advancement in sensor technology and human-computer interaction, our research aims to bridge the gap between computer science and health experts by building a personalized model that can detect physical and mental state events and provide corrective action through biofeedback. For example, it is known that respiratory illnesses such as influenza and COVID affect our breathing directly, and their early detection can lead to a more effective intervention or treatment. Our research problem includes, number one, identify different types of breaths and classify various respiratory events, including sneezes, coughs, speech, and other abnormalities like hypoxemia or sleep apnea. Number two, predict obstructive or restrictive respiratory conditions. Number three, utilize accelerometer to enable human activity recognition. And finally, correlate breathing patterns to various physical or mental states.
So the sensor that we utilize captures the abdominal or the thoracic expansion of the chest cavity through a piezoelectric force sensor. Our wearable also has a triaxial accelerometer and PPG, all measuring at 25 Hertz. As this device is non-invasive and has a long battery life, a participant can wear one tack throughout an entire day, allowing us to collect data 24/7 in the participant's everyday setting. We conducted tests to measure noise and eliminate drift algorithmically. So to discuss our methods a bit, we first need to talk about breath segmentation. We take a close look by segmenting the sequence of breaths into individual breaths based on their behavior. We then fit the lines over the waveform to capture the dominant shape of each breath. Our approach is to observe and analyze each individual breath. So as we see here, the segmentation is done and we can see a couple of breaths segmented with their inhales and exhales. And now we can discuss the breath features. Some of the breath features include the duration, which is defined as the total time of the breath. The amplitude is defined as the maximum value in the breath signal, which serves between the transition between the inhale and the exhale. This inflection point describes the height of the breath. Finally, the inhale and the exhale periods defined as the range of the X values for each separated by the height. Now Faizan will discuss the conclusion.
Thank you, Rifaa. In conclusion, and to describe our preliminary findings, we have developed precise online and offline segmentation algorithms, allowing us to create unique feature representations for each individual breath. Notably, per-breath analysis revealed that significant variability, underscoring the role of inhalation and exhalation patterns in event detection. The sequence of breaths tend to follow a cyclical pattern, which highlight the correlation between the minute-to-minute depth of breathing, meaning the amount of inhale that a person takes, and customized health models, because while breaths look similar, the significant variations from person to person, including differences in height, timing, and the inter-breath intervals, meaning the distance between each breath, highlight the need for a tailored approach in health monitoring and interventions. To describe our future work, number one, assess normal breaths under various activities. Number two, detect deviations from normal. And finally, correlate deviations to health conditions. Thank you for joining us.
This study investigates breath VOCs in type 2 diabetes, which is a disease for which the associated blood metabolites are already well established in literature. The purpose of this study is to demonstrate the capability of the Breath Biopsy Omni platform. Through our investigation of breath VOC profiles in type 2 diabetes, we aim to identify volatile metabolites associated with disease progression, in addition to compounds for which the association between blood metabolites and type 2 diabetes has been established in literature. Type 2 diabetes is a complex metabolic disorder defined by continuation of elevated blood glucose levels above 126 milligrams per DL at fasting. This is a disease related to long-term trends such as high calorie intake, associated changes in the gut microbiota, and lack of exercising. Type 2 diabetes is progressed from prediabetes and requires continuous therapy with drugs and blood glucose monitoring to ensure control and prevent development of associated comorbidities such as renal impacts. Breath is a complex matrix containing VOCs from several potential sources. Volatile metabolites can be released directly by cells in the lung or exchanged between blood and inspired air in the alveolar space. VOCs from the latter represent metabolic processes occurring at distal sites, including liver, gastrointestinal space, heart, muscle, and brain. To date, the challenge of breath sampling for the field is the development of standardized, sensitive, and reproducible methods to quantify breath-based VOCs. Our objective here was to evaluate VOCs in exhaled breath that differ in abundance among type 2 diabetes, prediabetes, and healthy control subjects utilizing Breath Biopsy Omni, a platform developed by Owlstone Medical and optimized specifically for the confident identification and analysis of compounds from exhaled breath. The goal was to use the platform to identify breath VOCs for which the association between blood metabolites and type 2 diabetes has been established in literature, in addition to new potential breath biomarkers. A total of 76 volunteers participated in this study, with gender and age well balanced across different groups, as presented in Table 1. Breath biopsy samples were collected using the Rifaa breath sampler, shown in Figure 2, which was developed by Owlstone Medical, and analyzed with TDGCMS using the Breath Biopsy Omni platform. VOCs that were not repeatedly present in breath samples were removed from the analysis. VOCs that were semi-consistently detected but have some missing values were assumed due to observations below the detection limit. The missing values for a given VOC were imputed with 80% of the minimum value of that specific VOC. With that, a little more than 1,000 compounds were moved forward for identification and statistical analysis. Compound identification was carried out in accordance with the MSI standards. Tier 1 compounds are validated IDs with reference standards run either in the same sequence or equivalent sequence. Tier 2 compounds are putative IDs assigned through comparison to Owlstone Medical's HRIM library. Tier 3 compounds are tentative IDs assigned through comparison to the NIST library. To investigate whether there was clear separation between different groups, PCA was utilized to visually inspect the underlying data structure. Figure 3 shows the result, which suggests a modest metabolic differences in breath VOCs among the different groups, as there was no obvious separation observed. Despite the modest differences in data structure, we found some interesting results with disease relevance in our further analysis, which focused on a subset of subjects that fasted over 8 hours, which also reflect a more accurate reflection of disease-associated metabolic changes. To determine whether the breath profiles in different groups could be utilized to train a predictive model for diagnosing diabetes, 3 different prediction models were tested. Shown in Figure 4, the inputs of these models were the concentration of significant VOCs in subjects with over 8-hour fasting, BMI, hours since the last cigarette, and hours since the last meal. In comparison to baseline accuracy, all 3 models performed well, with the decision tree exhibiting the best performance, achieving an average accuracy at 80.4%. In this study, a total of 156 VOCs reached statistical significance with unadjusted p-value below 0.05 between type 2 diabetes and healthy control groups. And a total of 96 VOCs reached statistical significance among the 3 groups. To identify biologically relevant VOCs, compounds that were obviously not on breath due to high backgrounds were excluded. Compounds without a consistent trend among the 3 groups were also excluded, as type 2 diabetes is a progression from prediabetes. This narrowed down the VOCs to those that can be roughly categorized into compounds related to microbiome, inflammation, and exposure. The key findings on selected VOCs are presented in Figures 5 and 6. Acetic acid and isovaleric acid are both short-chain fatty acids produced by gut bacteria and have been shown increase significantly in diabetic subjects in previous studies. 3-methyl-indole is a VOC in the tryptophan pathway, and compounds in this pathway like indole and indole are known to be relevant to type 2 diabetes, putatively modulating glucose metabolism. Methylpropanol ketone has been found to be associated with several diseases such as NAFLD and UC. Although the actual mechanism is still unknown, the increased levels found in type 2 diabetes in this study may suggest disruption in metabolic processes and inflammation. Additional VOCs found significantly increase in type 2 diabetes, as shown in Figure 6, include the tentatively identified gamma-anticholectone, 3-pyridinol, and an unknown compound. The biological relevance of these three compounds remain unknown. In conclusion, the prediction models in this study suggest that the significant breath VOCs found are capable of distinguishing different cohorts. The differences in exhaled breath VOCs among type 2 diabetes, prediabetes, and healthy control potentially reflect interesting physiological associations to the complex metabolic disorder of diabetes, such as metabolic processes, inflammation, the gut microbiome, and exposure. The findings indicate that the Breath Biopsy Omni platform is capable of detecting disease-associated metabolites already established in the literature, in addition to new potential breath biomarkers. For future metabolic disease studies, a larger number of participants fasted overnight will ensure better baseline status. Similarly, a population of treatment-naive subjects will serve as a better cohort for identifying biomarkers for disease diagnosis and prediction.
Hello everyone, I'm Dr. Anup Gurung from Department of Chemistry, SRM University, Sikkim. And today I'm going to present, this is my second presentation in the Breath Biopsy Conference. And our work is fabrication of porphyrin-based colorimetric sensor for the detection of liver cirrhosis biomarkers. Here in this study, what we have done is we tried to detect the biomarker of liver cirrhosis, which is in our latest research done in many parts of the world, especially in Owlstone group. It is found that the liver patients cannot process the D-limonene, which is primarily found in citrus food that we take, and the people with liver problem, they cannot digest it very well. All right. So cirrhosis is one of the D-limonene is one of the biomarker in which can tell about the condition of the liver. So we have designed a sensor which has the unique optical properties, that is, they are different colors and they are porphyrin based. Porphyrins are nothing but like hemoglobin is also a porphyrin based compound which contains iron and a porphyrin ring in it. Chlorophyll is one of the compound which is green in color, which has a magnesium inside. In this field, there's seen many people like T. Shukaram et al. Etc. They have shown that the limonene is not digested properly by the people with liver cancer or liver cirrhosis, and it is present in their breath in quite high frequency. This is the structure of D-limonene, which is present naturally. So what basically we are doing is we are sensing this particular molecule through our sensor and how much it is contained in the breath of the patient. So in our lab, what we have done is we have synthesized and characterized the porphyrins and its metal derivatives. First, we have synthesized, but in our study, we have tested 6 sensory elements. One is free base, that is without any metal. This is called free, without metal free. And when you put zinc, iron inside, this is iron porphyrin, copper porphyrin, conin. One source is lemon and another with sweet lime. And to see whether this is a blank experiment that we have done with water, and to see whether it like how the blank experiment goes, whether in the blank experiment also it changes color or not. So when we did with the water, so RGB, so there are 6 slides present here. So before exposure, we have exposed it to the, before exposure we have measured the RGB value, this one, and after exposure we have RGB value, this one. So we have subtracted this from this and we have got exposure value of this. So for 6 slide, when we expose to water, the RGB changes not much, it's 724. So when you put this value into the values, the color you get is all black. That means there is not much change of color. So 2 minus 2 is 0, which is black, almost 0. Okay, so this is a RGB change. So same thing when we expose those another set of slides with the lemon, which contains the biomarker. In some cases, in most of the cases, the RGB difference is pretty high, and when we put the value of these two to the RGB value, we get this kind of change color. Okay, this is a representative. This is when you put, so this is a difference map. It's called difference map. All right. Now, from here we can say that there's a particular kind of pattern is coming up. Okay, so for particular source also it is giving particular pattern. When we use the sweet line, the color changes like this. Okay, so it proves that the sweet lime and lemon mostly contains the biomarker D-limonene. It's showing a appreciable change in sensory element compared to only the water. Data analysis. So it was found that there is a considerable RGB difference between before and after exposure to limonene from sweet lime and lemon, but not in blank experiment like water. Free base TPP cup water vapor. So free base TPP, copper and manganese TPP has shown less RGB difference compared to, so iron, zinc and cobalt is showing appreciable. So thank you all for having the patience. And these are my references that we have used. Thank you all.
In the study, we investigated the allergen-induced acute lung inflammation in asthmatic individuals using Excel Breath. Here we use the allergen challenge model, which is considered the gold standard for investigating airway inflammation, and it is used, for instance, by pharmaceutical companies to test the ability of new drugs to reduce inflammation. In our study, 9 asthmatic individuals were randomly assigned into allergen or diluent exposure group, followed by washout period of approximately 4 weeks. During each exposure, Excel Breath was taken at baseline and 7 different time points, as indicated in the method section. The Excel Breath was collected using Breath Biopsy system and volatile organic compounds analyzed using state-of-the-art gas chromatography coupled to high-resolution Orbitrap system. The analysis consisted of targeted compounds, as indicated in Table 1, as well as untargeted, that is, all compounds that can be detected in Excel Breath. As can be seen in Figure 1 and Table 2, the allergen challenge induced changes in airway function as seen with reduced FEV1 and change inflammation measured in blood and sputum samples of allergen group. In the first step of our analysis, we evaluated the differences between breath profiles and ambient blanks. This is particularly important to know if the signal in Excel Breath is coming as a result of metabolic changes or only inhaled from the ambient. In Figure 2, PCA score plots are shown with Excel Breath samples indicated as orange cycles and ambient samples represented as gray cycles. As can be seen in both panels of Figure 2, Excel Breath and ambient samples create separate clusters. indicated that volatile organic compounds measured in exhaled breath have meaningful origin. In the following analysis, we have performed cross-sectional analysis to evaluate significant changes in exhaled breath volatile organic compounds with respect to baseline values between the diluent and allergen challenge at every time point. We use sign rank test due to paired nature of the data, and the volcano plot of the results is shown in Figure 4, with several compounds showing significant changes and/or large fold change. In addition, we perform longitudinal analysis to evaluate overall differences between the two exposures, and we use linear mixed FL models to capture dependency over time. In Figures 3, the box plots of selected two compounds demonstrating clear changes in their concentration over time between diluent and allergen are shown. In the last step, the correlation analysis of the air wall function and inflammation measure was performed and are shown in Figure 5. As can be seen, several volatile organic compounds that have been associated with allergen challenge showed mostly positive correlation with airway function and inflammation measure. To summarize, the current study shows that there are significant changes between allergen and diluent at different time points. Moreover, the present study shows that the allergen challenge induces inflammation in the lung and can be associated with changes in volatile organic compounds measured in exhaled breath. Several volatile organic compounds related to inflammation show significant correlation with blood and/or sputum measures. This may provide valuable insights into the underlying biological processes and interactions between the immune system and the metabolic pathways. With respect to the biological origin of the volatile organic compounds, we can relate them to ROS and inflammation as seen with elevated amounts of aldehyde and ketones.